Bismillahirrahmanirrahim. Assalamu alaikum. Today I am going to talk about a very common technique used in biochemical or protein engineering lab, the polyacrylamide gel electrophoresis, or we call it PAGE. And it is just a modified method for the gel permeation chromatography, and it is oftenly used in any type of biological lab. And when you will be in biological lab, then you will use it frequently. And we often use in this technique to identify amount of protein present, or its purity, or how many protein or how many types of protein is present in a sample of mixture, or what is the molecular weight distribution of my crude protein sample. And then we can identify this easily by this process. In electrophoresis chamber, we just set two different kinds of gel. One is stacking gel and another one is running gel. Stacking gel is just used for the sample loading, where the separation is not occur, just it is help the protein to migrate from the same position. And in the separated gel, the protein is get divided into groups, and protein with required buffer was added there. And the loading of the protein sample was done in very how we could say very careful manner, because it is really important not to mix up the protein from one to another well. You are seeing that this is very nearest well, and it is separated by very thin layer, so we need to be very careful when we add the protein here. And whenever we add the protein there with the micropipette, we need to be very careful that we do not add any bubble or anything which can take away the protein from the well and mix up too well, and then it will give us the wrong result. Now one need to, when you are very careful to do that, and you will able to put the protein or your DNA sample in each well like shown in this figure in video, then we will start the gel electrophoresis. And if we consider our chemistry, then it is nothing but a simple electrochemical cell where protein molecule run towards the negative pole as a total surface charge of protein and DNA usually negative. In addition, for SDS-PAGE, we add SDS, which is an anionic surfactant, and it is a well-known anionic surfactant used in molecular biology. And it not only help the protein to be linearized, but also make the surface completely negative, and which help the protein to migrate across the gel from negative electrode to positive electrode. And this migration basically benefit us. How? Because this migration help the gel to separate the protein molecule based on their molecular weight, or the separate the DNA molecule based on their molecular weight. Why? Because the size of the protein is now the driving force by which the protein or DNA will travel. And as we use the same gel and with the same percentage of acrylamide, which is responsible for the pore size of the gel is uniformly designed with the same pore size. So only one force or only one factor affecting here is the size of the protein or size of the DNA. And on the other hand, if we want to check the dimerization, multimerization, then we avoid use the SDS here and any other agent which can linearize your molecule. And if we allow our protein to run in that case in the native phase, then it will be also separated based on its molecular weight. Now what will happen? In the first case, the molecular weight was for only one molecule, because we use SDS, any other detergent, to detach them from, to remove their any kind of interaction with one molecule to another molecule. Even intramolecular interactions we just break down to make them linearized. But in case of this native phase, we do not do that. If we do not do that, so we will be able to determine the dimerization or multimerization or single protein, what is the state of the native protein, by this SDS-PAGE. So something we can do like with this SDS-PAGE or any other page. First of all, find out the molecular weight distribution. Second, molecular weight of the target protein. Third, whether the protein is purified or not. Fourth, whether the protein is monomer or multimer. And in case of DNA gel, we also have the ability to identify the molecular weight. Either my DNA is cut down in the same position or not, either it is ligated or not, either what is the weight of my DNA, what is the how many base pair could be there. And what we do, we just compare this line, the left side line, which is the marker. We know the molecular weight there or the base pair there, and just compare with this one, that oh okay, it is around. And it is not accurate, but it is the very much effective way to find out the protein molecular weight and also the molecular weight of our DNA or something like that, base pair of the DNA. And this will be very much essential for you, and this technique will be very much effective for you, and you will run this method every day in your lab. And hopefully this video will help you, and when you will run, you will learn more. And thank you very much. Hope this video will help you to learn the electrophoresis, and definitely you need to practice in your lab, then we will learn more. Thank you very much.